Okay. So we're going to talk about the nervous system and talk about its natural defenses. And then we're going to talk about diseases that are microbial in origin that affect the nervous system. So the nervous system is made up of the central and the peripheral nervous system. Okay. The sensory division of the nervous system is what brings stimuli in to the central nervous system, and that's part of the peripheral nervous system. And then the autonomic and the somatic motor divisions of the nervous system are part of the peripheral nervous system, and they carry commands out from the central nervous system. All right. The central nervous system is the brain, the brainstem, and the spinal cord, and they are well protected. They're surrounded by bone, and then they are surrounded by three membranes called meninges. And they're also bathed in a fluid called cerebrospinal fluid, which is a filtrate of blood, which is created by astrocytes and ependymal cells that line hollow cavities inside the brain and the spinal cord called ventricles. And in the spinal cord, it's called the central canal. And this allows the organs to float so they don't rest against hard surfaces and get damaged. And the fact that the cerebrospinal fluid is filtered by these glial cells means that it's going to be free of anything in the blood really that's infectious, unless you break what we call the blood-brain barrier, which is created by the ependymal cells and the astrocytes. So let's note that, right? The blood-brain barrier filters out pathogens and makes CSF, cerebrospinal fluid. And the cells that do this are the astrocytes and ependymal cells. Okay. So normally, bad stuff should never get into the central nervous system. And the same is pretty much true for peripheral nerves too. Okay. They are insulated and protected. Peripheral nervous system is protected by glial cells, such as Schwann cells and satellite cells. Okay. It's also protected by endoneurium, which is connective tissue. Okay. Perineurium, which is connective tissue. And epineurium, which is connective tissue. Okay. There's a blood supply that usually runs with the nerves and provides nutrients and removes waste products and provides oxygen, again, through diffusion and through the action of glial cells. Okay. And the result is that the nerves, the peripheral nerves, and the central nervous system organs really never touch blood, which means that the immune system really doesn't know they're there. And that's one of the reasons why, if there's trauma to the central nervous system, or if you slice a nerve, the immune system actually moves in and starts to destroy the nerve tissue as if it were an invader, because it's never seen it before. So it'll start tearing it up and start laying down scar tissue. And that's gonna cause loss of function in the central nervous system, and could cause loss of function in the peripheral nervous system, depending on the nature of the injury. So in terms of immune function, protection against infection, the only thing we really have for the central nervous system is a population of cells that are called microglia. Okay. These are glial cells that can phagocytose. Okay, so they can eat dead cells and they can eat pathogens, but if you get a big time infection, the glial cells are not enough to hold it off. Okay, and that's why the blood-brain barrier over here is so critically important. Got to have it, right? Got to lock those organs away from the blood supply and let the glial cells determine what comes into contact with the neurons so that they can continue to do their work. So in a sense, the blood-brain barrier picks out the nutrients and allows the oxygen to diffuse through, and it locks out everything else. Okay, so as long as there's no trauma to that barrier, everything's fine. Okay, the cerebrospinal fluid is constantly made from the blood and reabsorbed back into the blood at the level of the arachnoid granulations, which are little protrusions of the arachnoid mater into the dura mater that allow diffusion of the fluid back into the blood. Okay, so you're constantly making it and you're reabsorbing it, and so that flushes bad stuff out from around the central nervous system structures. Okay, the functions of the nervous system are sensory, integrative, and motor. Right, sensory is to turn stimuli, which are physical changes in, on, and around you, into electrical information. That electrical information goes into the central nervous system and gets basically meaning assigned to it, right? So we understand what information is coming in. And this happens for conscious stimuli at the level of the brain. Sorry. Stop it. Hold on. Brain. No. Oh, well. Happens at the level of the brain for conscious stimuli. And the brainstem and hypothalamus for unconscious stimuli, which is actually, believe it or not, greater than 90% of all stimuli. Right? So what reach our conscious thought is something around 5% of the stimuli that we experience. You might think, my God, we're missing out on a lot. But most of what you're missing out on is either for autonomic function, okay, or is what we might call noise, meaning it's not really important to us surviving, okay? Autonomic function is for homeostasis. Okay. The motor commands that come out are either from the autonomic nervous system or the somatic motor division. Okay. The somatic motor division targets only skeletal muscle. And the autonomic division targets everything else. And it is involved in homeostasis. Okay. This is voluntary movement. Okay. So we pick up the changes in our environment, we pipe them into the central nervous system, and the central nervous system interprets them, and then it generates a command and sends it out through nerves to what are called the effectors, the organs that are going to do the job that needs to be done. And that's the end of the loop. Okay. Neurons are dense structures. They're found in the brain and the spinal cord, and they're also found in peripheral nerves. We talked about the fact that the brain is surrounded by bone and meninge and cerebrospinal fluid. Okay. While the spinal cord's inside the vertebral column with the same meninges around it, the same cerebrospinal fluid. Okay. The peripheral nervous system is cranial and spinal nerves that are bundles of axons. They receive and transmit the nerve signals. The cell bodies of what we call the efferent division, the efferent divisions, are in the gray matter of the spinal cord. Okay. So they're getting their nutrients through the blood-brain barrier, right? The afferent division has cell bodies in the dorsal root ganglia, which are outside the central nervous system, and they get their nutrients through satellite cells, that take it from blood. Okay. And the oxygen comes through diffusion, right? And the blood vessels are very nearby. So we've protected the nervous system by and large from whatever's in the blood that could be toxic or could cause disease. Okay. So the only way something's going to get through is through trauma, right? Which would be like injury or maybe enzymes that can dissolve some of those cells away. So trauma, or if it's very small, or if it's non-polar, if it's lipid soluble, okay, then it can get in, okay? So very small would be things like tiny viruses, right? Non-polar would be stuff like oxygen and CO2 and things like steroid hormones, right? Those kind of things can slide through and get into the brain. Mercury metal can get in there, okay? Lead can get in there. So those are the kind of things that can slide through and get into the nervous system and bother it, okay? The way that the communication occurs in the peripheral nervous system is through what's called a chemical synapse, where the nerve cell has a long extension called an axon that stops right before the target, and then these little endings come off of it that are kind of like little swellings. Okay, they're called axon terminals, or sometimes they're called synaptic boutons, which is French for button. And they release something called neurotransmitter into the synapse, and then that gets sprayed onto the target. The target has receptor for the neurotransmitter, and that causes an electrical response in the target. Okay, either the target will do something or it won't. Okay, and so that determines the one-way nature of the nerve pathway, right? It's always going to go in one direction. It's not going to back up and go in the other direction. So we've talked about the blood-brain barrier, and we've talked about the fact that because the central nervous system isn't exposed to the blood, it has immune privilege, right? The possibility, for instance, of transplant of, say, nerve stem cells into the brain from any other individual is possible because of the fact that the blood never comes into contact with it, direct contact. Okay, there's only a few parts of the body that have immune privilege. Okay, it's found in the cornea, found in the liver, and it's found in the central nervous system. Okay, everything else, you need an organ transplant or tissue transplant, you have to match the antigens at or above 85% in order to have a chance for the transplant to take. Okay, otherwise the immune system will move in and destroy it. Microglia are phagocytic, and they're like the only immune defense in there. Okay, they clean up dead tissue, and they can eat pathogens, but if there's a major exposure, they're not going to be capable of knocking all that stuff out. Okay, so what sort of microbiota is there in the nervous system? Okay, the answer is none. Okay, herpes viruses can live in latency in the nervous system. They're not considered normal microbiota, but they're kind of cheating their way in, right? They're coming in through nerve endings by pretending to be something that the nerve cell needs. And then through retrograde transport, they're hitching a ride on motor proteins, and that's trucking them back to the nucleus. And then the genetic material just kind of stays in there, okay, and rests until it wants to be used again. Okay, but that in a sense is not a living pathogen, right? It's just information about a pathogen that could become active. But in terms of virus particles or endospores or cysts in normal central nervous system or peripheral nervous system, you just don't see them. Okay, so nothing there, right? So we're going to talk about some of the diseases that hit the nervous system. We'll talk about polio and meningitis and encephalitis, okay, and tetanus and botulism, okay. So meningitis just means inflammation of the meninges, okay. Anything that ends in itis means inflammation. Okay, and the meninges are just the protective membranes around the central nervous system. Okay, so a lot of different microbes can cause this, okay, and cause the same set of symptoms. Serious forms are caused by bacteria that get into the central nervous system, usually by co-infection or previous infection with respiratory viruses, okay? So the virus is coming in and damaging the blood-brain barrier, doing so, and that opens the door for bacteria to follow. All right. Meningitis in neonates is often caused by microorganisms in the birth canal, okay, versus what causes it in children and adults. So the way to figure this out is what's called a spinal tap. All right, this is a very painful procedure. Okay, a low gauge needle which means a big needle, a wide needle, okay, is inserted below L2 into the subarachnoid space. You got to be awake while they do this, which stinks, okay? It's below L2 because the spinal cord stops at L2, okay? You don't want to take a chance of touching the spinal cord, damaging it, okay? And then we check for bacteria, gram stain or culture, okay? A telltale sign is cloudy CSF. Okay, that means something's growing in there and it shouldn't. Okay. We treat with broad spectrum antibiotics immediately. Signs and symptoms: headache, stiff neck, fever, nausea and vomiting, photophobia, skin rashes, large number of lymphocytes in the cerebrospinal fluid, which means that we have done what? We've broken the blood-brain barrier. Okay. Neisseria meningitidis is a gram-negative diplococcus. Okay, so if it's gram-negative diplococcus, it's going to look like two little pink round balls, one right next to the other. Okay, gram-negative diplococcus. Okay, sometimes called meningiococcus. Associated with epidemic meningitis, causes the most serious form of acute meningitis. And there's 12 different strains out there that have capsules on them, okay? So the capsule is a virulence factor. Okay? And it probably keeps it from being eaten by macrophages. Serotypes A, B, and C are responsible for most infections. So let's take a look at meningitis. This is what we call bacterial meningitis. Okay.
Neisseria meningitidis, often called meningococcus, is a gram-negative diplococci that can cause meningitis and life-threatening sepsis. Neisseria are fastidious organisms that require nutrient supplementation to grow in laboratory cultures. Specifically, they grow on chocolate agar and can be isolated on Thayer-Martin agar, sometimes called BPN agar. Thayer-Martin agar is an agar plate containing vancomycin, polymyxin, and nystatin, along with other nutrients that facilitate the growth of Neisseria species, while inhibiting the growth of other organisms. Characteristically, Neisseria are oxidase positive, and Neisseria meningitidis can be distinguished from Neisseria gonorrhoeae because Neisseria meningitidis ferments both glucose and maltose, while Neisseria gonorrhoeae only ferments glucose. This organism exists as normal flora in the nasopharynx of 5 to 15% of adults and is spread through the exchange of saliva or other respiratory secretions. Virulence mechanisms include lipooligosaccharide (LOS) on the outer membrane of Neisseria meningitidis, which acts as an endotoxin and can cause shock and destruction of RBC. Other virulence mechanisms include a polysaccharide capsule that is antiphagocytic and aids in the evasion of the host immune response. Symptoms can rapidly progress from fatigue to meningitis with fever, headache, neck stiffness. If toxic levels of the endotoxin reach the bloodstream, meningococcal sepsis can develop, causing blood vessels to rupture and vital organisms to rapidly shut down. Complications include Waterhouse-Friderichsen syndrome with adrenal insufficiency, organ failure, low blood pressure, disseminated intravascular coagulation with widespread petechial rash, and death. Treatment includes ceftriaxone and rifampin prophylaxis.
So you can see here, right? These are the species for bacterial meningitis. We've got up here, Neisseria meningitidis, Streptococcus pneumoniae, and Haemophilus influenzae, okay? And these are always present in your body, okay? But they're kept at bay because of the normal flora and the normal defenses, right? Transmission through droplet contact, okay? So it's airborne, okay? Okay. What are its weapons? What are its virulence factors? The capsule that keeps it from being eaten by white blood cells, right? The endotoxin, all right? And an IgA protease. So what does this do? This destroys antibody. Okay. What does strep have? It has capsule. It can induce apoptosis, which is cell death. It has hemolysin, so it can rupture red blood cells. And it can produce hydrogen peroxide, okay? And then for Haemophilus, we have the capsule, all right? There is a vaccine for Neisseria. There are vaccines for strep, and there's a vaccine for Haemophilus, okay? If you contract this and you have the out and out disease, we have antibiotics. Okay, these are all antibiotics. Okay. Some strains have become resistant to some of these antibiotics. So there are reports that tell us which strains have become resistant to certain antibiotics, and they suggest other antibiotics to use. All right. And then what do you see in people that have this condition, right? So these would be signs. Okay. Petechiae, which are little blood spots on the skin or in the eyes. Okay. Meningiococcemia, meaning what? Bacteria in the blood. Okay. In strep, okay, you see the same symptoms, except here it's going to be streptosemia, right? And they're usually going to have adults affected. And then Haemophilus is less common because of the vaccines that are available, but you're going to see the same set of symptoms, right? Always petechia, and you're going to have bacteria in the blood, right? But because we have vaccines for these, okay, we can prevent this problem. Okay. So we can wipe out the bad player before he gets a chance to get in there and cause problems. Okay. So that was meningitis. These are some more meningitis candidates. Listeria, Cryptococcus, Coccidies, and there are viral meningitis candidates as well. So here the vehicle is food, so you ingest it. Here again, it's airborne. Okay, same here, airborne. Okay. What are the weapons? Well, Listeria can grow inside white blood cells. Cryptococcus has that capsule again. Okay. Coccidioides, all right, can form spherules, okay, and resist white blood cells. Okay. And here we have lytic infection of host cells, meaning for viruses, we can rupture infected tissue. Okay. Prevention. Here, cook your food and don't use unpasteurized dairy. Okay. Same here. Here, avoid the endospores. Okay. And here, we don't really have anything other than a vaccine. Okay. So that's what we're working on for some of these viral meningitis species. Treatment. Okay. These are antibiotics. Okay. So these are antibiotics. Okay. Cryptococcus. All right. This is an antifungal. Antifungal. Okay. And this is an antifungal as well. Okay. Right. So these guys are fungi, right? And for virus, usually nothing unless there's an antiviral for the infection or if there's an existing vaccine. Okay. What do you see? For Listeria, asymptomatic in a healthy adult, meningitis in the neonate, and the elderly. Okay. For Cryptococcus, it can be acute or chronic meningitis and often seen in AIDS patients who have immunosuppression. Okay. And here we see endemic breakouts for coccidioides. And this is usually milder than the bacterial or fungal types. Okay. Okay. For Streptococcus pneumoniae, okay, this is a gram-positive flattened coccus that is a diplococcus. It's often found in the nasal pharynx, causes most bacterial pneumonias, and is the most frequent cause of community-acquired meningitis. A quarter of the patients with pneumococcal meningitis will also have pneumococcal pneumonia, and it often occurs in patients with other susceptibilities. Okay. It has a polysaccharide capsule that protects it against the white blood cells. They produce alpha hemolysin and hydrogen peroxide. These damage the central nervous system and also damage red blood cells. It's capable of causing brain cell apoptosis, all right? So it can kill nerve cells or glial cells through chemical weapons. It can penetrate the respiratory mucosa and gain access to the blood and get into the meninges. Okay, so you have to remember here that the nose, right, this is the nasal cavity, there's a nostril. On the top of the nasal cavity, you have the first cranial nerve. Okay, the olfactory nerve is a real short collection of nerve endings that dangle into the nasal cavity and allow you to detect odor, all right? And what can happen sometimes is if you inhale virus, V for virus, okay? See, no, I'm sorry, bacteria. This is bacterial meningitis. So if you inhale bacteria, okay, then it can penetrate the respiratory mucosa and it can get into either these nerve endings, okay, or into the blood supply around the nerve endings, right? And from there, it can gain access to the meninges, all right, which are going to surround the entire central nervous system. And that's what it will infect, okay? Penicillin is not a good choice because the bacteria can be resistant. Ceftriaxone is used, but you have to make sure that the infectious agent is susceptible. It's also recommended that a steroid be given 20 minutes prior to antibiotic therapy in order to reduce the inflammatory response to cell wall components that are released by the antibiotic treatment. Okay. The two vaccines are Prevnar and Pneumovax. Okay. Haemophilus, prior to the vaccine in '88, was a common cause of meningitis and death. Now it's less common in the US because of a vaccine. It's a gram-positive bacterium that ranges from Coxobacillus to long filaments in Palisades. So Coxobacillus, you're going to see, and it's gram-positive, so it's going to be blue-purple, right? You're either going to see dots or rods or something in between, okay? A range of shapes in between, right? It can be in chains, or it can be in palisades, right? Palisades look like chandeliers. Okay. No capsule, and they have flagella on them too. Okay. Listeria, subclinical in healthy adults, but in the immunocompromised, the brain and the meninges can experience inflammation and septicemia, which is bacteria in the blood. A death rate of 20%. Pregnant women are susceptible because the microbe can get across the placenta or postnatally through the birth canal and get into the baby. Intrauterine infections can result in premature abortion and death. This is a gram-positive bacteria that ranges from Coxobacilli to long filaments in Palisades formation. So again, it's going to stain purple, okay? And then long filaments in Palisades formation, okay? So what you'll see, right, are parallel filaments that dangle like chandeliers. Okay? And again, there's going to be flagella on them as well. So they are motile. It's not fastidious, meaning grow easily on standard media. It resists cold and heat and salt and pH extremes, okay, as well as the detergent action of bile, right? It grows inside host cells, so it can grow inside a white blood cell. It can move directly from the host cell to another healthy cell, right? The reservoir is soil and water, okay? Animals, plants, and food can be secondary sources of infection. Most cases of listeriosis are associated with ingesting contaminated dairy products, poultry, and meat. It's in 15% of ground beef, 30% of chicken, and 10% of luncheon meats, hot dogs, and cheeses. Okay. Cold enrichment isolates the organism. So you put the specimen at 4 degrees and plate it onto media. This takes around 4 weeks. Rapid diagnosis using ELISA and immunofluorescence. Okay, so these are antibody methods to detect the organism. Okay, so we throw this magic bullet at it that has either a dye attached to it or something that glows when you hit it with a laser, and then or an enzyme, and we get a color, right? And we know that it's there, it sticks. Okay. Prevention, antibiotic therapy as soon as listeriosis is suspected. First choices are ampicillin and trimethoprim sulfamethoxazole, and you can also use erythromycin. Prevention is improved by pasteurization, proper washing and refrigerating and cooking of foods suspected of being contaminated, and pregnant women are cautioned not to eat soft, unpasteurized cheese. Okay. Or Cryptococcus. It's a chronic form of meningitis, has a gradual onset, and is often found in immunocompromised patients. It's sometimes called meningoencephalitis. You see headache, stiff neck, nausea. It's a fungus that has a spherical shape with small constricted buds and a large capsule. Primary area where you find this is in the bird population, in areas where you find pigeons. It likes high nitrogen environment in the bird droppings, okay? Birds are unusual because when a bird goes, all right, he's hold on. So when a bird makes bird droppings, okay, what it is it's a mix of urine and feces, okay, and it comes out through the cloaca, which is a common opening for what would have been the urethra and the anus. All right, so a bird just has one opening there. All right, and because there's a urine component to it, that's high nitrogen. Okay, because urine has got a lot of nitrogenous waste in it. Okay, so the Cryptococcus likes that. Masses of dried yeast cells are scattered in air and dust. Healthy humans are normally resistant to it. The high rates of infections are in immunocompromised patients where it's often fatal. We can use steroid treatment to cause the infection to get worse. Diabetes and cancer can also cause the infection to get worse. And all of these are going to suppress the immune system. Okay. How do we prevent? We start with amphotericin B and fluconazole over a period of weeks. And what are these? These are antifungals. Or we can call them antimycotics. That's the technical term for an antifungal. No prevention, unfortunately. Okay, coccidioides, causative agent, another fungus. It likes to grow at room temperature on moist areas. It generates a moist white-brown colony with a lot of branching hyphae, which are the little fuzz things that go into the media and also are above the surface of the media. Okay. On special media at between 37 and 40 degrees, which is around body temperature, the arthrospore germinates into a parasitic phase, which is a spherical cell called a spherule. There's two species, okay? Imidis is responsible for diseases in San Joaquin Valley, and Posatocil is in the Southwest, as well as Mexico and South America, okay? So it's endemic. What is it that makes it cause disease? It has high virulence. It begins as a pulmonary infection that can go all over the body. All persons inhaling the arthrospores probably develop a degree of infection, but some are genetically susceptible. The arthrospores become spherules, okay? Spherules. Mild respiratory symptoms can develop into disease, including meningitis, okay? Southwest is where this usually happens, 100,000 cases annually. We also see it in Central and South America. Concentrated reservoirs are in the San Joaquin Valley and in northern Arizona. A 94 outbreak was traced to Northridge after an earthquake loosened clouds of dust that had spores, and winds carried them to residential areas, okay? Aseptic meningitis is the type that is produced without bacteria or fungi, okay? This causes 80% of the meningitis cases, and it may not have an infectious cause. Often these occur in children, 90% by enterovirus. Herpes simplex 2 is a common cause. Occasionally other viruses can get in. It usually resolves within 2 weeks, so it's much milder, and a lower mortality rate at below 1%. The diagnosis begins with the failure to find pathogen in the cerebrospinal fluid under light or fluorescent microscopy, and we confirm it by viral culture and antigen tests. In most cases, no treatment is needed because there is no treatment, okay? There's really no treatment for this. The virus has to run its course. Neonatal meningitis is almost always the result of an infection transmitted from mother to child in utero or during birth. As more preemies survive, the rates of this meningitis go up. It's a favored condition in patients with an immature immune system. So again, what we're seeing here is suppressed immunity. Okay, that's the doorway, right? Most common causes: strep algisate, E. coli, and listeria, okay? The Streptococcus algolacti causative agent is a group B streptococcus that colonizes 10 to 30% of the female birth canal. Prevention and treatment: penicillin, sometimes with aminoglycoside. Women are high risk, and they're usually screened between 35 to 37 weeks into the pregnancy. When we detect it, IV antibiotics are given at the beginning of active labor, and erythromycin is given in the case of an allergy to penicillin, Okay, so it's a streptococcus, right? So it's going to be chains of bacteria. Okay, one next to the other, next to the other, next to the other. Okay, chains of little round cells. Okay. E. coli, it has a K1 strain, the second most common cause of neonatal meningitis. It occurs in preemies with a 20% mortality rate. And even with aggressive treatment, those who survive have permanent brain damage. Prevention, usually transmitted through the mother's birth canal. And we can use antibiotics such as ceftazidime or cefepime. Gentamicin can be administered through IV. Coronobacter, okay, formerly known as Enterobacker, is found often in outbreaks of neonatal and infant meningitis, transmitted through powdered infant formula, mortality rate 40%. The FDA and the CDC have told hospitals to use ready to feed and concentrated liquid formulas versus powder. They also advise hand washing and cleaning feeding equipment and using fresh formula and discarding anything that's left over. Okay, so here we see the deal with meningitis. All right, so these are all bacteria. All right. The virulence factor here for strep is a capsule, right? Which means it resists phagocytosis. Okay. Here we can grow inside white blood cells. Okay. And here we can survive dry conditions, right? E. coli K1 is a special strain because it contains a lysogen, okay, that generates a toxin that causes the damage, okay? So this is sort of a mutated form of the bacteria that normally lives in your gut. Again, what do we treat with? Okay, all of these are antibiotics, okay? Antibiotics. Okay. So we'll break here for 15, and we'll come back for the last half of this, okay? So I'll see you guys at 10 after, so 2:10. Okay. Okay. Poliomyelitis is a viral disorder that is largely in the past now because of the development of the vaccine. There were two vaccines, an oral and an injectable. And one of those was developed at the University of Cincinnati, actually. So let's talk a little bit about this old disease of the nervous system. It's an enteroviral infection in the spinal cord that can cause neuromuscular paralysis. So what happens is it gets in through the digestive system. It also used to be called infantile paralysis. A campaign greatly reduced the incidence of polio, but there's almost no civilization on the planet that has gone unaffected. Okay. In 2014, the World Health Organization declared a public health emergency because polio had spread to 8 countries in the Africa and the Middle East. It's been widely eliminated in the Western world due to the prevalence of the vaccine. Okay, so let's look at poliomyelitis and how it's contracted and how it's spread.
So how does the polio virus cause poliomyelitis? So how does it infect a person and end up causing the symptoms that we see, which is muscle damage, muscle paralysis, that sort of thing? Well, first of all, the poliovirus is what's considered an enterovirus. Enterovirus. And an enterovirus is a sort of virus that likes to replicate in the intestines. So this little virus will end up getting into the mouth of the patient, will be swallowed, and get into the intestines. Once it's in the intestines, it'll replicate, it'll duplicate itself until there's more and more virus in the body. Now, this enterovirus, poliovirus, can actually replicate in the throat as well. So you see these two sites of replication. And once it has replicated to a certain number, so there's a large amount of the virus in the body, the virus is somehow able to spread to the central nervous system. So it actually gets to the spinal cord, right? Here's the spinal cord right here, and attacks the cells of the spinal cord. Actually, specific cells of the spinal cord, which I'll get to in a moment. But how does it actually get to the spinal cord? Well, that's actually not entirely known. There's two theories of how it can get there. One is viremia, which is the virus gets into the blood. Emia refers to anything associated with being in the blood. So the virus gets into the blood. And then from the blood, it somehow finds its way to its specific target in the spinal cord, these neurons in the spinal cord. So that's one theory. The other theory is that there is something called retrograde axonal transport. Retrograde means it goes backwards, and it's transported backwards. And axonal refers to the axon of a nerve. So generally, what a nerve looks like is a cell body, right? This is where the nucleus, the cytoplasm, and the other organelles or aspects of the cell that allow it to grow and survive and all of that jazz. You know, every cell has a cell body. But what's special about neurons is they have this long tail, or this little long pathway that we call an axon. So this axon is how nerves, which are collections of these neurons, transport information. They send electrical impulses down the axon. And at the terminal of the neuron, there's some sort of chemical reaction that occurs to pass this information on to, you know, perhaps muscles of the body or potentially even to other nerves. So the idea here is the poliovirus is somehow able to get into this axon and somehow travels up to the cell body where it does its replication and damage of the neuron. So it's postulated that neurons serve as a highway from the site of replication to the spinal cord where they do most of their damage. Just to recap, poliomyelitis is a motor neuron disease. So motor neurons are the target of the poliovirus. Now, really quick, people who are infected don't necessarily show symptoms. In fact, 90 to 95% of people are asymptomatic, even after being infected, and they usually recover. However, even patients who are asymptomatic, who are infected by polio, can still transmit this virus and shed it out of their intestines through their feces. In fact, people can be asymptomatic, not have any symptoms, but shed this disease for up to 4 to 6 weeks. So the virus spreads and continues through the population. So you can see that even though many people recover and don't experience symptoms of polio, at some point it can get to somebody who will experience the symptoms. And it's really kids about less than 6 months old that are most at risk. And the problem is these kids, they wear diapers, and in areas of improper sanitation, this virus can just get spread all over throughout the community. Now, taking a step back to the pathophysiology, I have this cross-section of the spinal cord drawn out because I want to emphasize something. The motor neurons are the cells that are targeted by polio, and they're in a part of the spinal cord, right? This is the entire spinal cord, known as the anterior horn. Anterior means in the front, and horn is just kind of the way it looks. So these are the cells in the front of the spinal cord. Now, just a quick recap of the spinal cord. This that I have outlined here is the gray matter. Gray matter is where the cell bodies are. This white surrounding area, known as the white matter, is composed of axons. So we've got the white matter here. So it's these cell bodies of the anterior horn, which so happen to be the motor neurons of the body, that are affected by the poliovirus. So you've got all these little neurons, and they connect to different muscles of the body. So if I show it on our little guy here, there's neurons that are all throughout this spinal cord, neuron cell bodies, and they connect to different muscles. And here let's draw it in this purple color so you can see it a little bit better. So these neurons right here are connecting to the biceps. Now, when polio kills some of these neurons, might kill some of them, but not all of them, you have decreased output to the muscles. So let's say these neurons are killed off. You no longer have innervation or support provided to the muscles. They're no longer getting this electrical output from that neuron. It's been killed off. So patients will experience weakness and not be able to use their muscles effectively. So the symptoms you're seeing are from damage to these motor neurons. And sometimes they're actually referred to as the lower motor neurons. Remember, there are connections between neurons. So there are neurons in the brain that connect down into the spinal cord, and then they connect with this other cell body of a neuron that goes and provides innervation to the muscle. So what I was talking about over here is saying there's a connection to another cell body of a neuron. Okay, so there's this lower motor neuron disorder and damage. Thankfully, there's a way that the body can compensate. Even though this neuron right here might be damaged, right, it's no longer able to provide any input to the muscle. Other neurons, which have axons that extend to that muscle too, may sprout additional axons to provide innervation to the area where the old neuron used to provide innervation. So this single neuron here is now controlling more of the muscle movement. So some patients get compensation and recovery from polio through this re-innervation of collateral sprouting, as it's called. However, as a patient gets older, they can experience something called post-polio syndrome. And this occurs years and years later, after the initial acute infection with polio. Post-polio syndrome is defined as onset of functional deterioration. So the muscles are getting worse after a long period of stability. So even after this recovery, years later, the patient may continue to experience weakening of the muscles. Now, why does that happen? Is the infection staying in there and continuing to damage other neurons? No. What's actually happening is through the aging process, these neurons just start to die. This is the normal aging process. You lose function of neurons. Now, normal people have a lot of different neurons serving their muscles, so they don't experience this weakness as severely. But since you have now maybe only a few neurons that are trying to provide a lot of support to a single muscle or muscle group, when these cells die just through the normal process of aging, a patient will experience continued and worsening weakness of the muscles that were originally affected. So neurons all throughout the body are dying, but it's these neurons that are having this additional extra effort to support the muscles that when they go, the patient really starts to notice that weakness. So again, post-polio syndrome can occur years after the original infection of polio.
So you can see here, again, right, it's a virus. So what do we do about it? And the answer is a vaccine, okay? So vaccine. Okay. What can we do for treatment after infection? Nothing. Okay. We can deal with pain, but there's nothing we can do once it's in there. All right. And the virulence factors are the ability to attach virus to GI cells and enter neurons. Okay. So that's the weapon. Okay. Meningoencephalitis caused by Nigella fowleri and acanthamoeba. Okay. These are parasites that invade the body under unusual circumstances. So these are protozoa. Okay. They are eukaryotes. Okay. With Nigella fowleri, it's an amoeba. It's found in warm, natural bodies of water. It's forced into the nasal passages. All right. It burrows into the mucosa and migrates into the brain and surrounding structures following the olfactory tract. Okay. You know this better as the brain-eating amoeba. Okay. It causes primary amoebic meningoencephalitis, massive destruction of brain and spinal tissue, causing hemorrhage and coma. Okay. Death within a week. What is it doing? Well, it's burrowing. Okay. And it uses enzymes and phagocytosis to kill tissue. Okay. Bad way to go. Wide distribution in fresh water, very common. Children carry the amoeba as harmless biota during the summer months. The events that lead to infection are rare. Okay, so this is something that is what we would call, I wouldn't call it opportunistic, but I would call it accidental. Okay. Naegleria fowleri It advances rapidly. The treatment basically usually isn't in time. Early therapy with amphotericin B, sulfadiazine, or tetracycline can be of some benefit. And again, these are antibiotics. Okay. No general means of control is out there. Swimming pools and baths have to be chlorinated and check for the amoeba. Okay, so even after you chlorinate it, you have to look for it. Acanthamoeba, okay, similar to Naegleria, right? The infection course is longer and it causes something known as granulomatous amoebic meningoencephalitis. It gets in through broken skin, through the conjunctiva, and sometimes through the lungs and the urogenital epithelia. People with traumatic eye injuries and contact lens wearers and AIDS patients exposed to contaminated water are at risk. Okay, so we're dealing here either with trauma or immune suppression. Okay, so you can see here the different types, right? These guys are protozoa. Okay, and so the treatments, right? These guys are antiparasitics. Okay. What are the virulence factors? What are the weapons? Highly invasive, okay? Meaning can penetrate normal barriers with enzymes and phagocytosis. Okay. Acute encephalitis. It can be either acute or subacute. This is inflammation of the brain. Okay, so itis, inflammation, right? And encephalo, referring to the head or the brain. Okay. It's a serious condition. Tissues of the brain are sensitive to the inflammatory process because they have never been exposed to the immune system. Okay. Acute encephalitis caused when the viruses are borne by insects, such as West Nile or herpes virus. Bacteria can cause encephalitis, but the symptoms are more pronounced in the meninges. What do we see? Behavior changes, confusion because of the inflammation, decreased consciousness, seizures, meningitis symptoms. We do treatment with acyclovir if it's viral. Okay, this is an antiviral. Okay. Treatment won't harm people infected with other agents, right? Most arthropod vectors feed on the blood of the host, and peak incidence is during the late spring and early fall. Mammals, are going to maintain the virus in the cold and dry season. Humans are a dead-end accidental host or a maintenance reservoir, depending on the virus. The impact, millions of people get it each year, thousands die annually. One common outcome is fever and rash. Symptoms and management are caused by arbovirus are similar. Transmission and the range of infection of individual viruses are different, okay? So what are the weapons? What are the weapons here? Okay. Hang on it. Hold on. Yeah. There we go. Weapons. Okay. Insect bite. Virus gets into the tissues and reproduces in white blood cells. Right? So it uses them as host. Okay. All arboviruses are mosquito transmitted. Prolonged viremia gets the virus into the brain. Inflammation and swelling damage the brain, the nerves, and the meninges. It's variable for the symptoms. It can include coma, convulsion, paralysis, tremor, loss of coordination, memory deficit, changes in speech and personality, and heart disorders. Survivors can experience permanent brain damage. Young children and the elderly are sensitive to injury, okay? Detecting arboviral infections, you have to have a patient history. You've got to do serum analysis. So this is going to involve antibodies, using antibodies, okay? Serological tests for some viruses detect antibody. Prevention, acyclovir. So this is an antiviral. Okay. Support measures controlling fever, convulsions, dehydration, shock, and edema. Control safeguards are aimed at controlling the insect that carries it. Carries the virus. So you can see here the distribution and some of the notes for the different types. Okay. Herpes simplex 1 and 2 can cause encephalitis in newborns and HSV positive mothers. Prognosis is poor. Older children and young adults are susceptible, as well as older adults. Okay. Encephalitis is a reactivation of dormant HSV in the trigeminal ganglion, usually in these older children and adults, right? So this means, okay, reactivation of dormant HSV. This means that it was latent. Okay, so resting inside the DNA of your own nerve cells. Varicella zoster can also reactivate from its dormant state and can be responsible for rare cases of encephalitis. Varicella zoster normally is the cause of chickenpox and shingles. Okay. JC virus got its initials from the patient that was first diagnosed with it. It's a polyomavirus. It can cause progressive multifocal leukoencephalopathy, okay? It develops in patients that have immune suppression. They're immunosuppressed. Okay. It's generally fatal in those that get it. What happens is they attack accessory brain cells, meaning what? Glial cells. We get demyelination of parts of the cerebrum. A few deaths have been prevented with zidovudine, an antiviral. Okay. So you can see here, acute encephalitis, right? So these are spread by vectors, okay? These are spread by reactivation of latent vector, okay? Or by vertical transmission. That would be from mother to child, for instance. And JC virus is probably everywhere, including in our own DNA. We normally don't see it because it is controlled by our immune system, right? The virulence factors for arbovirus encephalitis are attachment, fusion, and invasion, right? The ability to get into a host cell get inside host cells and reproduce. Okay. Subacute symptoms take longer and are less striking. Causes include toxoplasma, which is a protozoan. Or measles virus, okay? Or prions, which are toxic proteins that are normally consumed in infected meat. Okay. So let's look at toxoplasma.
Toxoplasma gondii is a protozoan parasite of the phylum Apicomplexa. It is distributed worldwide and can infect all warm-blooded vertebrates, where it develops as an obligatory intracellular parasite. Its final host is the cat. With feces, the cat sheds environmentally resistant oocysts. These sporulate outside the host to infectious stages. For the development, called sporulation, humidity, temperatures of about 20 degrees Celsius, and sufficient oxygenation are required. Inside the oocyst, at first, two sporoblasts are developing. Within the next 2 days, they become sporocysts. They are now surrounded by a wall and each contain 4 sporozoites. Having entered the intermediate host, excistation occurs in the lumen of the intestine. Under the action of intestinal enzymes and bile salts, first the oocyst is open. Here we see one isolated sporocyst still wrapped in the ruptured oocyst wall. With digestion of the sporocyst envelope, the parasites are eventually set free. Once more, the process of excistation. The parasites are termed sporozoites and are the organisms infectious to all intermediate hosts. Sporozoites are formed as elongated cells of 7 by 2 micrometers. They show active gliding motility where the parasites are gliding on a glass slide. All single zooites, sporozoites, and, as we shall see later on, tachyzoites are similarly structured. The zooide contains a basally located nucleus and other organelles, just like any other eukaryotic cell. A typical feature is the apical complex. It is composed of a conoid and 3 types of secretory organelles. Small vesicles, the micronemes, are believed to play a role in host cell recognition. The rhopteres are elongated club-shaped organelles, 8 to 10 of which are found in each zooide. The third kind of secretory organelles are the dense granules, which will play a role after the invasion of the host cell. Infection of intermediate hosts occurs from oral uptake of oocysts containing sporozoites. Raw or undercooked meat containing tissue cysts loaded with invasive organisms named bradyzoites is a further source of infection. Once in the host, the parasite crosses the intestinal cells and starts multiplying in various cell types such as macrophages, fibroblasts, and neurons. At this stage, which is the acute phase of the disease named toxoplasmosis, a transplacental transmission is possible. This can be fatal to the fetus. The invasion of host cells can also be observed in vitro in tissue cultures. The invasion of host cells is based on active motility. Parasites glide on glass slides, on the cell surface, and even within the host cells. They always glide on a substrate. The mechanism is not yet fully understood, but involves an actomyosin motor located beneath the parasite membrane. The occurrence during the invasion will now be shown in detail. Having reached the host cell by gliding, the apical contact triggers signals that lead to the exocytosis of micronemes. Their contents create a stable bond between the parasite and host cell. The junction is then moved posteriorly. At the parasite apex, a vacuole forms, which develops further with the apical discharge of lytic substances from 1 or 2 of the rhopteres. The vacuole membrane remains continuous with the host cell membrane throughout the process, but no host cell protein diffuses into the vacuole membrane. The vacuole membrane is a composite between host cell membrane lipids and rhoptere contents. After internalization, the dense granules move to distinct exocytosis sites to be discharged into the vacuolar space. The released material accumulates and is gradually associated with contorted tubular structures termed the vacuolar network. Dense granule and rhoptere proteins associated with the parasitophorous vacuole membrane are believed to participate in the organization of transmembrane pores. These may contribute to the nutrition of the parasite and leave free access to molecules up to 1500 Daltons. Finally, host cell endoplasmic reticulum and mitochondria accumulate around the parasitophorous vacuole. Host cell invasion observed here in tissue culture only takes a few seconds. Apical contact is followed by smooth gliding inside the cell. The constriction at the site of entry can be seen during the whole process. Electron microscopy would reveal the moving junction here. After invasion, the parasite begins with its intracellular development. This is shown in a tissue culture of human fibroblasts. This is a close-up of an early vacuole. A major feature of the vacuole is that it never fuses with any other compartment of the host cell endomembrane system. Within the vacuole, the parasite begins to multiply. Multiplication is by repetitive binary fission and is referred to as endodiogeny. It is a very complex process where, after nuclear division, two daughter parasites form inside the parent organism. They will eventually emerge from the parent, which will gradually disappear, being progressively incorporated into the daughter zooids. Repetitive multiplications produce further parasites. They are called tachyzooids. Each division takes approximately 6 to 12 hours, depending on the strain. The fastest growing parasite is usually the most pathogenic in vivo. The vacuole grows in size and will eventually occupy most of the host cell volume. It finally reaches a stage when the host cell cannot physically cope with further parasite development and ruptures. The egress in this case has been artificially triggered by calcium ionophore. Free tachyzooids will then disseminate throughout the entire host and infect new cells. Active invasion also occurs in macrophages, which are a most favored host cell in vivo. In this case, the process is identical to what was shown before in fibroblasts. A moving junction is formed and invasion is completed within a few seconds, just as in any other cell type. When the host starts generating an immune response, tachyzooids are efficiently eliminated by macrophages. This mechanism of host defense, called phagocytosis, can be shown in cell culture. Phagocytosis is not oriented apically as with invasion. The parasite is surrounded by the cytoplasm of the macrophage and incorporated. This process lasts more than 1 minute. After phagocytosis, the parasite is killed and digested. Escaping parasites are able to maintain a chronic infection. These parasites developing in cysts are termed bradyzooids and grow mainly in muscular tissue. The cysts can persist for years. They are usually controlled by the host's immune response and remain quiescent within the tissue without causing any harm to the host. However, when the host suffers immunodeficiency, such as in AIDS or upon organ transplantation, the infection can be reactivated. Bradyzooids can be released and switch back to the tachyzooid stage. This acute phase can be fatal in humans. Cysts are the main source of transmission to final or intermediate hosts. A cyst is surrounded by a thin wall, which is made by the parasite as a deposit under the membrane of the parasitophorous vacuole in the host cell. Cysts can be isolated and aggregated by gradient centrifugation. The host cell is destroyed in this process. Bradyzooid release can be triggered by proteolytic enzymes such as trypsin, just as occurs in the gut of a carnivore after eating infected tissue. First, the bradyzooids invade enterocytes, later other tissues. There they multiply as tachyzooids, resuming the intermediate host cycle. Ingestion in the intestine of a cat leads to the invasion of enterocytes, where the bradyzooids undergo a typical coccidian cycle. In the epithelial cells of the duodenum, multiplication or schizogony takes place. The resulting merozoites invade neighboring cells. They develop first into macro and micro gametocytes, then into macro and micro gametes. This is known as gametogenesis. By fusion of the flagellated microgametes with the macrogametes, an oocyst is produced. Oocysts are excreted unsporulated. That means that they contain a single cell or zygote. The thick oocyst wall protects them against external influences, and is extremely resistant to disinfection. It can persist infectiously for several months. Inside the oocyst wall, sporulation takes place. It lasts for about 30 hours and is shown here in time-lapse. With this development phase, the life cycle of Toxoplasma gondii is completed. After oral uptake of these infectious stages, the cycle can begin again.
There's prions.
There is evidence that the infectious agent associated with certain brain diseases, such as Creutzfeldt-Jakob disease in humans and mad cow disease in animals, may be caused by a protein. The infectious agent has been called a prion, for proteinaceous infectious particle. The normal prion protein, PRPC, is bound to the surfaces of neurons, and the gene for PRP is present in normal vertebrates and invertebrates. Presumably, the normal prion protein, PRPC, can be altered, and the altered prion protein, PRPSC, SC meaning scrapie associated, becomes misfolded and takes on a different conformation. Many researchers believe that the infective agent is this alternative protein conformation. It is believed that when PRPSC enters a normal brain, it binds to the normal cellular PRP, PRPC, and induces it to fold into the abnormal conformation. The newly produced PRPSC molecule may then convert more normal PRPC proteins into the abnormal PRPSC form. An alternative hypothesis is that PRPSC could activate enzymes that modify the PRPC structure, not shown.
Prions are hypothesized as the causative agents of several neurodegenerative diseases. Prion diseases are unusual because they appear to be caused by an infectious protein. The proposed mechanism of the disease is that the infectious...
Okay, that one's not showing. So you can see here, again, the different causative agents, right? So this is subacute encephalitis, and our suspects are Toxoplasma, all right, which is a protozoan. Okay. We've got measles virus, okay, is obviously a virus. Okay. And then we have prions, which are infectious proteins. Okay. So what are the virulence factors? Well, growth for the protozoan inside white blood cells and neurons. Okay. For the virus, cell fusion and evading the immune system. Okay. So a lot of these are going to be enveloped viruses. Okay. And then the prion avoids the immune response because of its small size and its location, right? Which is in the central nervous system. Okay. We only need one bad particle to get in there and set off that chain reaction where it misfolds all the other proteins that causes the disease, and it basically turns the central nervous system into gelatin in just a few days. Okay. You know this better as a mad cow disease. Okay. Okay. Treatment. Okay. These are anti-protozoals. Or if you like, anti-parasitics. Okay. Nothing here for the virus. Okay. and nothing for the prion. All right, so you get it, it has to run its course. Okay. Let's take a look at rabies. Here. Oh, come on. Hold on just a second.
Hi, I'm Peter Costa with the Global Alliance for Rabies Control. Today I'm talking about rabies, a deadly yet preventable disease that infects the brain. Now I want to talk about the common symptoms of rabies. If you think that you have been bitten by or exposed to a rabid animal, it is very important to get medical attention as soon as possible, because rabies is almost always fatal, and once symptoms appear, medical intervention is rarely successful.
You start showing some of the first signs of rabies, which are very vague, flu-like. You're going to wake up in the morning, you might have a fever. You might have some aches and pains. You just may not want to get out of bed. It's going to be experienced for a couple days. Thereafter, a very short period of time, you're going to get into a very acute neurologic phase. What does that mean? That means you're going to have hallucinations. That means you might feel tingling at the site where you were bitten at. You may not be able to sleep. You may start salivating to the extent that you need several boxes of tissues to wipe that up. Very shortly thereafter, you're going to have problems breathing. You're going to have problems with your heart. And shortly thereafter, you're going to go into a coma, paralysis, and death. All of that happens over about 14 days. Prevention of exposure and prevention of disease before clinical signs are what it's all about. Because if you start showing signs of illness, you've bought the farm, it's over, it's too late.
During this time, medical treatment is ineffective and sedatives are administered to help comfort the patient. The first sign of rabies in an animal is usually a change in behavior. It is important to remember that while many health conditions will cause a pet to have a change in behavior, rabies is a progressive disease, so the health condition of the animal will only get worse until it dies. Animals that have rabies usually stop eating and drinking and may seem to want to be left alone. Early signs may include a change in the tone of the animal's voice because the rabies virus infects the voice box, chewing at the site of the bite, fever, a loss of appetite, and other subtle changes in behavior. After the first signs of rabies appear, some animals become very aggressive, and bite other animals and humans or even inanimate objects. Animals may also become very docile and are difficult to wake up. Rabies in animals quickly develops into paralysis and ends in death. Based on public health information, we know that in some cases there is a low probability that an animal involved in a biting incident actually had rabies. In these particular circumstances, keeping the biting animal under observation and in isolation, where it can be observed for a period of 10 days, may actually rule out the need to euthanize the animal. If the animal that was involved in the biting incident was actually rabid at the time of the bite, it would begin to show signs of rabies and in fact would most likely be dead within the 10-day observation period. If you would like to learn more, check out our other videos on rabies, including diagnosis of rabies.
So you can see here some of the animals that can carry rabies. It's spreaded by the bite, okay? Color coded, right? Look at all the bats spread throughout the US, right? Skunks, raccoons, common vectors around here, okay? And if you head south, more raccoons, southwest, foxes, skunks, okay? We have coyotes in Ohio now, so you can count them in. So this chart probably needs a little bit of updating. Okay? So you can see here, the virus is going to use as its weapon an enveloped glycoprotein. All right, so this is an enveloped virus, meaning that it's disguised in host membrane. Right, that's its weapon. That's its weapon. Okay. And what is the treatment? Okay. Immunization, as soon as you bite, if you've had the bite, right? After it sets up, it's just what we call palliative, which means we deal with your symptoms and you eventually die. Okay. So it's to get it before the symptoms appear. Okay. Prevention, vaccine. There is a vaccine out there. All right. Let's look at tetanus.
The tetanus vaccine is a little different than the other vaccines we've talked about, in that tetanus is a bacteria that's infectious, but not contagious. We're not going to get it from other people. In fact, we're going to get it from something as common as dirt. Now, it's not specifically the dirt. There's a bacteria that lives in it called Clostridium tetani. And the bacteria itself is actually very easy to kill. It's very sensitive to heat and to oxygen, but it has the capability of producing a spore, kind of like a seed, if you will, that can last for months up to even 40 years in dirt and still be able to cause disease. Now, it doesn't happen just by you touching the dirt. It happens from that bacteria being introduced into your body, typically with a deep penetrating wound that's dirty. Everybody always hears that you're supposed to worry about stepping on a rusty nail, but actually doesn't have anything to do with rust. It has to do with that nail was probably in the dirt. That's the reason it got rusty. Now, what happens is that bacteria gets into that deep wound and starts to grow. It then releases one of the most potent neurotoxins known to man that then spread through the bloodstream and the lymphatic, and then affects the neurotransmitters and allows muscles to go in unopposed contraction. Now, tetanus can happen one of three ways. It can happen through localized, which is actually fairly rare and very rarely lethal, in which you just have these uncontrolled spasms in the area that was affected by the wound. Another very rare one is cephalic tetanus, which usually happens with a wound that's in the face or around the ear. And this will just cause localized spasms of the face. But the most common is generalized tetanus, where you just have uncontrolled spasms of the entire body. It typically starts in the face and the neck and results in your jaw locking down. thus the common term for tetanus called lockjaw. Now, these spasms typically can start anywhere from 3 to 14 days after the wound happened, with an average of about 7 to 8. The spasms can last for minutes at a time and can happen from anywhere from 1 to 4 weeks, within resolution completely taking months. Complications include actual fractures of your spine or your long bones just from the severe contractions of the muscle. You can also have complications such as seizures, aspiration pneumonia, high blood pressure, and even death. The vaccine is available and given at 2 months, 4 months, 6 months, again at 12 to 15 months. A booster given between 4 and 6 years of age, 11 to 12 years of age, and every 10 years after that. Now, it's only good for actually 5 years for a dirty wound. So if you've had your vaccine within the last 5 years and you get a deep penetrating trauma, you don't need another tetanus vaccine. But if it's been longer than 5 years, we'll typically give you one at the time we see you in the doctor's, for the penetrating wound, or if you need stitches. Now, the vaccine has been very effective and has been given for almost 100 years now. So we've seen a huge decrease in the amount of tetanus in the United States. Infants can actually get tetanus through infected umbilical cords. This is very rare in the United States, but actually still fairly common in the world, causing over 250,000 deaths per year in infants, usually around the time they're 14 to 21 days of age. So it's important to get this vaccine, because the common myth of, I'll just stay away from sick visits, doesn't work when you're talking about dirt.
So you can see here how the tetanus toxin does its dirty work, right? The bacteria get in through a penetrating wound. They generate the tetanus toxin. The tetanus toxin ends up spreading into the central nervous system and can affect the motor neurons, right? And what that does is it causes the excessive firing of excitatory impulses to muscle, and that's going to cause damage to the connected bones. It can also result in respiratory paralysis. Okay, so all of this is very dangerous, which is why, again, the minute you get a wound, right, you want to make sure and get the antitoxin. Okay, same with rabies, right? The minute you get the bite, you want to get the antitoxin in you before the symptoms begin. Okay, if not before, right? So that's the organism, right? Clostridium tetani is a bacteria. Okay. Okay. Transmitted usually through contact with a reservoir, primarily soil or water. Okay. And the weapon is this exotoxin, right? This is what causes the disease. So it impacts nerve function. Causing spasm. Okay. The treatment, the antitoxin. Okay. Or immunization prior to exposure. Okay. After exposure, antibiotics. Okay. And muscle relaxants are to deal with spasm. Okay. Okay, let's take a look at botulism. Doing this again. Hold on.
We're going to discuss.
So, sure. Sure.
We're going to discuss botulism in this video. And botulism is a very interesting condition that can occur mostly in pediatrics on the licensing exams, but it can also occur in other populations. So what is it? The specific type that we're going to discuss is Clostridium botulinum. That's the name. And this releases a toxin, and that toxin is responsible for muscle paralysis. And I'll explain why, but I just wanted to quickly tell you the very brief summary. So what does that mean, muscle paralysis? Well, think of it as the opposite of muscle contraction. Muscle contraction can't happen. Muscles can't contract because of this toxin. Now, very quickly, before I get into this topic, I wanted to talk very quickly about something called Botox. And I'm sure many of you have heard of it. It's a very popular cosmetic procedure. It's a medication that people have injected into their skin to treat wrinkles. Now, what's that all about? Well, what Botox essentially is the botulinum toxin. So wrinkles are basically muscle contractions, right? So if you give Botox, you sort of do the opposite. You make these muscle contractions, which are wrinkles, paralyzed, and that's why you get rid of the wrinkles. Very interesting way of taking something that's bad and marketing it to do something good. Anyhow, so let's get back into Clostridium and the botulinum toxin. So why is this happening? When you have the botulinum toxin in your body, what happens? Well, very quickly, without going into too much detail, I wanted to draw a muscle cell and show how it reacts, or not reacts, but interacts with the neurons. This is a neuron, this is the axon. Now at this end, you have the release of a neurotransmitter. And ACH, as many of you can probably figure out, is acetylcholine. And acetylcholine is a very, very important neurotransmitter. And it's involved in proper muscle function. It goes out from the neuron, and there's these acetylcholine receptors on the muscle cell that catch it. And the muscle then is able to contract. So the acetylcholine neurotransmitter essentially is involved in proper function of the muscle. What botulinum toxin does is it blocks this. It blocks the release of acetylcholine. So you don't get acetylcholine and coming out of the neuron and interacting with the muscle cell. So if that happens, that leads to the inability of contraction. So the muscles can't contract. And when the muscles can't contract, you get something known as paralysis. And they give a special name, flaccid paralysis. And that is essentially the fundamental problem in botulism or botulism. So why would somebody get this? If somebody's normal and all of a sudden they develop botulism, what is the reason? Well, there's three ways. You can either get it through food, you can either get it through a wound, or a baby can get it.
Interestingly, the baby gets it from food also by ingesting honey. We'll talk a little bit about that later. Food, the most common type of food are canned foods. Canned foods, unfortunately, I'm not saying all canned foods have this, but canned foods can unfortunately carry this toxin. The good news is that if you cook the food at very high temperatures, it can kill the toxin. And in particular, what you're doing is you're killing the spores. And that prevents this from happening. So what are the symptoms? We talked a little bit about one of the symptoms, that's paralysis, probably the biggest muscle paralysis. And it also infects the respiratory muscles. And that actually causes respiratory failure. So it's actually a very serious problem if this occurs. The muscles of respiration are not able to properly work, and that can lead to severe problems. Constipation is another non-specific symptom. And there's other ones that they tend to list in clinical vignettes, like ptosis, which is drooping of the eyelid. And there's another one that's very, very commonly listed as a symptom, and that's floppy baby. They use this. Now, what does that mean, floppy baby? Well, there's a picture of this at the very beginning of the video. I don't know if you noticed that before you click play. Essentially what that means is a baby that has very poor muscle tone, hypotonia, and that makes it so that the baby can't really contract their muscles when you're trying to pull the baby up. And that's known as floppy baby. So how you diagnose this? Very difficult diagnosis, but fortunately there is a way to isolate the organism, which is Clostridium botulinum, in the stool, the toxin in the stool. And that's a very special test that's done in the laboratory if this is suspected. And the treatment, well, fortunately, there's something called an antitoxin to fight the toxin, and it's given a name, equine. This toxin, unfortunately, doesn't really, it doesn't inactivate the toxin that's already present, but what it can do is it can help or slow the progression of this condition. And basically the hope is that with time, the nerve endings are able to regenerate, but that can take weeks to months. Another very important part of the treatment is intubation because the respiratory muscles are involved. So let's take a look at some vignettes. 8-month-old baby presents to the ER with 1-day history of poor feeding and generalized weakness. The mother states that she often feeds the baby honey to pacify her. The toxin responsible for this presentation works by which of the following mechanisms? Well, if you rewind back in this video, you will see me trying to explain that this toxin blocks the release of acetylcholine from the nerve terminal so that it can't interact properly with the muscle cells, and that eventually causes muscle paralysis. And the last one, a 4-month-old infant presents with failure to thrive, progressive muscular weakness, and poor head control. On questioning, the mother states that she typically feeds the baby soy-based formula sweetened with honey. Which of the following organisms is most likely to be responsible for the child's presentation? I like this description, poor head control. That's referring to the baby's floppiness, floppy baby. I also like this question because it gives you 4 types of Clostridium. There's actually even one more. There's 5, I think. Now you might get confused by this and say, well, I have no idea. But interestingly, each types of Clostridium have their own disease and own symptomatology. And now this video, of course, we were talking about the first one, Clostridium botulinum. So that's the correct answer, A.
So you can see here the causative organism, right? Clostridium botulinum, the bacteria, right? Bacteria. Okay. What is the weapon? Botulism exotoxin, okay? And that causes paralysis. Okay. And then the treatment, antibiotic. Okay. And then antitoxin, which is antibody against Botox. Okay. And supportive care. All right. Now, what's the primary way this happens? You consume it. You eat it in the food, right? All right. So one of the things to look for instance, in canned foods is gas in the can. So the can looks plumped out. Okay. That means there's probably Clostridium growing in there. Okay. It's a spore former. All right. So it can sometimes slip by the sterilization process. Okay. And then raw foods. Such as honey and milk. Okay. No pasteurization. That means bacteria can be in there. That means Clostridium can be in there. And this is just a look again at the different diseases, what causes them, okay, specifically, and what group you would find the pathogen in. Okay. So a nice little chart, again, to help you with the diseases of the nervous system. All right. So now let's talk for a minute about the lab. All right. If we look at the lab, we're going to see here, and share this.
I can't see it, Mr. Connors.
I'm bringing it up.
Okay.
Here, back about big. All right. So again, here we are, right? We're in week 5, all right? And the lab is enteric bacterial identification, all right? So what we're doing here, let's do annotation with this. There we go. There we are, okay? So these are, move this over to the other window. There we are. Annotate. There we go. Okay, so yeah, so enteric means in the gut. Okay, so this would be the GI, the digestive system. Okay, and the idea, if we were in a live lab, is that we would look at some bacteria and check for whether or not they're anaerobic or aerobic. What conditions do they grow under? And we would start the culture in this lab, and then we would look at it in the next lab. So the media that's used to do that is something called thioglycolate broth. Okay. And with thioglycolate broth, what you have is a tube with a liquid in it, thioglycolate. And at the top of the tube, you have high oxygen. Okay. And at the bottom of the tube, you have low oxygen. Okay. And then what you do is you inoculate your bacteria into the tube. Okay, so you'd have different species of bacteria, and so you'd have different tubes. You would inoculate those different tubes, and then you would look for growth the following week, right? And so if the bacteria grew near the top, they would be aerobic. Okay. If they grew near the bottom, they would be anaerobic. Okay. If they grew throughout the tube, okay, they would be facultative, meaning they can switch off. They can use a variety of oxygen concentrations to do their work. So I'm going to send you a PDF and a website in the email that will explain how you'll do the lab. Okay. What you'll do is you'll visit a particular website, you'll do an activity, and then you'll fill out the PDF and then you'll email it to me. Okay. Just like you did a couple of weeks ago with the Gram stain. Right. And then I will grade that, and that will be this week's portion of your lab grade. Okay. And then I'm going to set up a practice midterm for you on Connect for you to do. And then we will have a midterm. Let's get out of this. We will have a midterm. Let's see here. Because week 4 technically is when you do all the midterms, but they're doing it week 5. So that's fine. I will have a midterm for you on both the lab and the lecture. Okay, the lab midterm is something that I'm going to have to cook up. Okay, and I may post it, I may send it to you. I don't know yet how I'm going to do that, but I will let you know. But the lecture midterm will definitely be on Connect, and there will be a practice midterm to go with it. And it'll be everything that we've done up through the end of lesson 5. Okay, so it's going to be 16 and 17, 11 and 15, 4 and 5 and 6. Okay, and 1, 2, 3, and 4. Right. So there'll be equal numbers of questions from each of those chapters. Okay, and I'm looking at probably 50 questions, 2 attempts. Okay, and it'll be due by the following week. All right, and then, like I said, I'll let you know on the lab midterm how I'm going to do that. Okay, I may do that.
Is that the midterm that's already posted on Connect?
Yes, that'll be the midterm already posted on Connect. But like I said, I'll put a practice midterm up for you so that you can practice with that before you take the real one. Okay, and there won't be
Do we
any
get credit for it?
Yeah, I'll put it in Connect, and there won't be any points associated with it. It'll just be like a drill. Okay, and you want to take that before you take the real midterm to see how you do, and then you know whether you're ready. Okay, and I'll try and do the same thing for the lab midterm if I can figure how to do that. Get you a practice. Let's see, can we clear all this? Yeah, we clear all that.
You didn't send us nothing for lab for this week, did you?
No,
'Cause I didn't get it.
no. All I sent you was that there was a lab in Connect for you to do on using the microscope to see what's in the pond water. Okay, and I think that was worth 10 points. So if you go on Connect, you'll see that there. You can do that. And I will send you the other part of that lab for last week. And I'm also going to send you the thioglycolate lab this week. Okay. So you do those, complete them, and just get them back to me, and then we should be cool. Okay. And of course, I'm going to send you the podcasts to all the lectures up to this point, so that you'll have that to study from as well. Okay. And remember
And you know,
to come to office hours if you want extra help, right? 5 to 7 on Fridays. Okay. Any other questions?
Never got a link for that.
I'm going to send that today. I'm going to send that today. There was an old link that I think needed to be replaced, but I'll send all of that once I've got the podcasts uploaded for today. Any other questions? We good? All right. I will see you all next week. Again, come to office hours if you have questions or email me, and I'll get you all that material. And if you didn't get it, or if you need old material, let me know and I'll resend it. Okay.
Okay.
All right. Thank you,
Okay.
everybody. Appreciate it.
Okay, thank you.
bye.